having us today.
Thank you. Thank you. Really excited to have you guys. And I'll turn it over to you to start our presentation for today.
Absolutely. Just before I turn it over to the real stars of the show, Richard and Jim, I just want to say thank you for having us. It's been a pleasure to work with CF Lifestyle. Over the last few years, we had the pleasure of participating at the Knockout CF event back when in-person events were possible, and look forward to the return of that event in the future. But I'm excited to be here. I work on our corporate communications team and oversee our patient advocacy efforts. So if you have any questions after the presentation, please don't hesitate to get in touch with us through our social channels as well. But without further ado, I'll let Richard begin the presentation.
Wow, thank you, Maura, and thank you for the invitation today to come and share with you first some background, but then some data on the work that we're doing in terms of developing mRNA therapeutics for pulmonary diseases, with cystic fibrosis being really front and center for us, and our lead program, the lead disease, which we're actually working towards with our therapeutics. As Maura said, my name is Richard Worcester. I'm Chief Scientific Officer with Translate Bio. I've been with Translate for 2 years now. Before that, I've spent 15 years in the pharmaceutical industry, and before that in academic labs in London and Cambridge in the UK. Before I jump into the background, Jim, maybe I'll just ask you to introduce yourself so we can have a smooth transition once we get to that point.
Sure, yeah, thank you, Richard. So my name is Jim Sullivan. I started relatively recently here at Translate, and I'm really excited. I came in to head the pulmonary discovery effort. Before joining Translate, I spent a long time at Vertex. I worked at Vertex for about 12 years, and that really represented a diversity of roles there that, at various times spanned really, really early research all the way through to late clinical development, and even included some opportunities to be able to go out and have community-facing discussions about CF. And I always found it to be so rewarding in presenting data from Vertex and really thinking about the science and the translational science that we were doing, but then really to be able to understand how they were actually leveraged in the clinic, and to be able to, talk to the people who were actually employing the medications, as well as an opportunity to meet some patients as well. So I had a brief period away from respiratory when I went and worked in cell and gene-based therapy, and I just had to get back to respiratory and CF. And so Richard presented the opportunity for me to come and join Translate. And I'm just really excited about the opportunity that Translate has now to take this platform that has been in development for a long, long time, and really recognize the value that it could have for patients.
Fantastic, thanks, Jim. And so before jumping into some sort of real specifics, I think setting the stage for you in terms of mRNA and how we see the potential for this in terms of therapeutics. So Maura, if you could move to the next slide. I think many of you will be familiar with this paradigm where we inherit the DNA from our parents, that encodes and how it contains our genetic information. And inside our cells, we have this process of transcription, which then reads the DNA to create the messenger RNA. And those instructions then move out into the part of the cell where proteins are made. And these proteins, not just the building blocks of our cells, but actually perform many, if not most, of the functions within our cells. And of course, these cells all build together to make us who we are. And of course, we know that there can be inherited defects in DNA, for example, in the CFTR gene in the case of cystic fibrosis. And so that sort of mutation, that defect in the DNA follows through this path, ultimately resulting in a defective protein, which then causes all the symptoms and the consequences of that defect in CFTR. Now, with a lot of existing small molecule drugs like Trikafta, really they're focused around the protein and how those small molecules combined interact with the proteins and change, modulate, if you like, the function of the protein. And that's fantastic if there's an opportunity for the protein to be made, or if there's protein, but it's not quite fully functional, fully effective. But when there's no protein, those molecules have nothing to bind to, nothing to work with. And so for those individuals with cystic fibrosis who are making no CFTR protein, really, we know that the modulators in most cases have really no effect. So what we've set out to do is to hit the center, hit the bullseye here in the middle of this slide with the mRNA. And so if we can deliver the functional mRNA, that can be translated and make the protein and restore that CFTR function. Now, you heard earlier that this sort of mRNA has come in perhaps into many people's sort of lexicon and understanding now because of vaccines. And let's look at things just briefly. I'll touch on vaccines in a few minutes again, but many of the existing vaccine technologies were using protein. And so a protein would be made in a manufacturing facility, and that's what would be injected into your arm or injected, and that's what would then cause that vaccine to be effective. Now, what we've seen over the past year, of course, is that mRNA vaccines for COVID-19 have been made, and now many tens, if not hundreds of millions of people have actually received an mRNA-based vaccine. And for us, I think an aspiration is to follow that and to have mRNA therapeutics really grow in the same way and be, I think, a mainstay of therapeutics in the same way that mRNA vaccines have done that in the infectious disease space. So how do we do this? And what are the components that can make an mRNA therapeutic? And that's shown on the next slide. So there are two building blocks that go into making our MRT platform. The first of those builds on that last slide. It's the mRNA itself. And there are these different sections of the mRNA which are critical to have an mRNA which is then going to create the protein. mRNA is a very unstable molecule. It can be degraded very rapidly. So we protect the mRNA in the structural sense by this 5' cap and the poly-A tail. And that protects that coding region in the middle. That's what codes for the CFTR protein. But even with those protections and with that mRNA, we need to deliver it to the cell where it's going to be expressed in the lung. And the way we do that is we package the mRNA inside a lipid nanoparticle. So here we've made these proprietary lipids that, when we mix them together with the mRNA, form this sphere, this ball-like structure. And what we do then is we put this into a nebulizer as a solution. That vibrating mesh nebulizer then creates an aerosol. That's what we're looking for patients to inhale. And then that lipid nanoparticle takes the mRNA through into the lungs and into the cells to make protein. So to show you how all of that comes together, we have a short animation, which is on the next slide.
Deep within cells, genetic information encoded within our DNA is transcribed into messenger RNA, or mRNA, which is needed to produce proteins, the molecules that perform essential functions in our bodies. mRNA sends a message to our cells with instructions to produce proteins through a process known as translation. Genetic diseases are caused by mutations in DNA sequences that result in mRNA providing the wrong instructions to cells. This leads to the production of dysfunctional proteins, or none at all. Translate Bio aims to treat diseases by addressing the central step of our body's protein production process. We design and optimize an mRNA sequence containing instructions for making the desired protein, and then package it into a customized delivery vehicle, the lipid nanoparticle. The lipid nanoparticle protects the mRNA from degradation until it reaches the target cells in the body, where it provides instructions to enable the cell's own machinery to make functional proteins. Translate Bio believes that mRNA technology can be applied broadly across many diseases where the production of a desired protein could provide therapeutic benefit. mRNA may also be used to make vaccines that prevent disease. In this case, the mRNA vaccine is intended to provide instructions for the cells to produce a viral antigen, causing the body to generate a protective immune response. Relying on more than a decade of mRNA expertise, our unique platform of mRNA design and delivery is at the core of our mission. Developing transformative mRNA medicines to treat or prevent diseases.
Oh, Richard, I think you're muted.
Sorry, I was muted there while the animation. This is a static version of that animation, just so you can recap on the different steps here. And as you can see, as the LNP is taken up by the cell, what we do in the labs is we actually optimize the different components here. So we make different lipids, we test different mRNAs, really to drive ultimately the protein expression, which you can see on the right-hand side of here with that ribosome, which is taking the mRNA that we've made and producing these proteins. And so we've built now a team who have the expertise to go through those different stages of optimizing the molecules. We test them in our preclinical models to assess whether they're effective or not. And that, of course, is an important step before we ask patients to work with us and actually start to evaluate these in clinical studies. We mentioned vaccines as well in that animation. And there are some important differences between what we do for our pulmonary applications compared to vaccines. Of course, in a vaccine setting, we're looking to make the antigen, for example, for the coronavirus. Whereas for cystic fibrosis, the protein is that natural protein that is not present in many CF patients, CFTR. And of course, with a vaccine, that's an intramuscular administration, whereas we have now built a strong group in terms of this inhaled approach to administering our LNPs. And with a vaccine, it might be there's only 1-2 doses, perhaps for life. We'll see what happens with the coronavirus, whether there needs to be annual boosters. But for cystic fibrosis and other chronic disease settings, we're expecting that these would be taken on a routine, regular basis by individuals to help restore, in the case of cystic fibrosis, the lung function. Now, you might be asking the question, well, how does mRNA compare to other types of genetic modalities? Because there are certainly other opportunities. And so what this slide is showing is that with mRNA, we're absolutely using the cell's machinery to make the protein. And if you could advance Maura to to build out the slide. So we're making the cell's own protein with mRNA, because gene editing, I think that's a real a goal which certainly is would be a great place to get to. We're still working through, I would say, in terms of how to do that and other aspects of where that could take us to. But with gene editing, you're making that change, and then if that's there, the change is made and can't be undone, can't be altered. With mRNA, what we see is we can change the dose, we can change the frequency. We can really work with patients on what's best and what's going to give them the best outcome. And so we're excited about mRNA therapeutics for now and where they're taking us. And what I'll do is hand over to Jim to describe from this theoretical application of mRNA now into real data and a real practical application.
That's great, Richard. Thank you so much. Maura, if you want to advance the slide, I just want to actually begin by reinforcing, some comments that Richard made, particularly where, we're all really excited here about, the fact that we have demonstrated, or that the community has demonstrated, the utility of mRNA-based vaccines. It's really exciting, but at the same time, that's obviously a different context than of thinking about mRNA as a therapeutic for a disease that is a chronic genetic disease. And so let's just take a step back. I know everybody here is aware, but CF is a monogenic disease. And so really what it means is that patients that have CF, have are homozygous for mutations within the CFTR gene, right? The gene that is producing the CFTR protein that's responsible for ion transport, specifically in particular chloride transport across the epithelium, and where the deficiency of CFTR in the lung is essentially the underlying cause of the following disease pathology that results from this genetic disease. But it's also worth pointing out at this point in time that this term monogenic means one gene, and there's an amazing amount of complexity that underlies that, which at this point I think everybody in the CF community is well aware of, given the fact that the different CFTR modulators have different indications based upon the particular mutational profile. So just to highlight that. And really, as we think about that and we advance to the next slide, Mara, when we think about what the goal of the promise of these mRNA therapeutics are, one of the key promises is the fact that it has the potential to reach all patients regardless of the underlying mutation. And again, as Richard points out, that also includes patients that have mutations for which a CFTR modulator would not be a viable mechanism because of the fact that little or no protein may be produced. And really, the opportunity then to deliver the mRNA really, as Richard also pointed out, means that it's an opportunity to treat the underlying cause of the disease rather than treating symptoms, downstream consequences of not having a functional CFTR protein. And so if we take a dive a little further and think about what that first line means in terms of the diversity of genetic mutations that can cause CF, this is a schema that I'm sure many of you are familiar with, where essentially what we're looking at now are the different classes of CF causing mutations, the different types of CFTR deficiencies that can present and cause CF. In these different classes, the CF research community has done a great job at spending a lot of time characterizing these different classes of mutation, understanding, the distinction between a protein defect like Delta 508, F508 del, which is this class 2 defect that results in a folding defect or trafficking defect, as compared with a mutation like G551D, the classical kaleido responsive mutation, where a protein is made and is able to make it to the cell surface, but is non-functional. And essentially, regardless of which class of mutation underlies the cause of disease, being able to have this therapeutic platform that's described on the left-hand side that involves delivery of a novel mRNA that can produce a full-length protein should affect the underlying cause of disease, regardless of which mutations a patient would have. And as Richard pointed out, that also does include this class 1 deficiency, where patients aren't making full-length CFTR protein. And when we start to think about how that relates to patient populations, and Richard already commented on this earlier, but if we kind of look at the distribution then of these different types, not necessarily on the next slide, Maura, of the different classes of mutation, but we now split these up into essentially therapeutic types, it really is this 9.4% of the population where a CFTR modulator is not indicated, won't provide benefit, where you could imagine, a really nice opportunity to be able to have this mRNA therapeutic platform that could essentially then replace the deficient CFTR protein, recognizing that there may also be some additional patients even that have mutations that are indicated for some of the CFTR modulators where patients may not be deriving benefit. So if we think about how this relates to our clinical application in the next slide, we do have, and if you want to advance one more slide, Mara. Yeah, perfect. So what we're going to do now is just kind of very briefly go over the Translate's first assessment of a CFTR mRNA therapeutic packaged in one of these lipid nanoparticles in a clinical trial. And really, the thing to point out here, one of the key aspects of this is really that this trial design reflected a first-in-human trial. And a key point there that I want to highlight is that the objective that's defined on the left-hand side is really to evaluate the safety and tolerability of this, particularly in CF patients. Can we actually deliver this LNP, this mRNA packaged within a lipid nanoparticle to the lungs of a CF patient, and will it be safe and tolerable? And in order to assess that, we looked at this in the context of a randomized double-blind placebo-controlled trial. And all that means is that, the investigators and patients didn't know whether they were getting a placebo or an active drug, so that we could make sure that the trial was run with all the proper integrity, and that the interpretations that we were drawing related to that objective of safety and tolerability were robust. And then one other piece that I want to comment on here for the results that we have reported so far that are defined in this blue box is that they incorporated both what we call SAD and MAD groups, where the groups on the left-hand side in part A reflect a single ascending dose, where patients were administered one dose at these different dose levels. And then ultimately, the safety and tolerability outcomes from those enabled the assessment of multiple ascending dose cohorts, where we're able to assess the safety and tolerability in the context of repeated doses up to 5 weekly doses. The last comment that I want to make about this particular clinical trial is that it's actually still ongoing. And so while we're talking about some some interim data based upon the shaded cohorts in parts A and B, that there are still cohorts that are ongoing that include a 20 milligram dose in a MAD cohort, as well as what's defined in purple, which is an effort in order to assess how daily dosing, the tolerability of daily dosing as compared with a weekly dosing regimen. And on the next slide, we're just going to highlight a really high-level overview of what our outcomes have been to date from this study. And just as a reminder, this study again was designed in order to assess the safety and tolerability. And as the first inhaled mRNA therapeutic, particularly, also the first to be administered via repeat dose, the main high-level finding so far from the interim analysis is that it has been generally safe and well tolerated. Lung function, which everybody here knows well, specifically percent predicted FEV1, was included as a safety endpoint. And to that end, it was not negatively impacted in those patients that were receiving the active treatment. But at the same time, there were no obvious patterns of increases in percent predicted FEV1 either. And then we think about two other parameters that we were really interested in this study. The first one was really understanding whether or not the presence of the CFTR protein, which would be generated from the mRNA that was delivered, whether or not that or the delivery vehicle itself could be immunogenic. And we tested that, and the conclusion is that there are no clinically relevant immunogenicity by looking at both anti-CFTR antibodies. Are we generating immunogenicity to the protein that we're delivering CFTR, or to part of the delivery vehicle, which is this anti-PEG antibody, as well as whether or not we see our body's immune response being activated to CFTR. And the high-level conclusion is that we're not seeing any of those patterns. And then the last piece is really, one of the key questions here, one of the key challenges, truthfully, which I think we all recognize as a community, is that when we're delivering a inhaled route of administration for any CF product, we don't have the opportunity to be looking at systemic biomarkers that could tell us what our levels of, whether or not we were hitting target levels of exposure, but also, as we're all used to in thinking about systemically delivered modulators, the opportunity to look at sweat chloride, for example, because we really are targeting delivery specifically to the lung. What we did decide to do, though, was to look in the blood to see whether or not we could detect any mRNA or lipid. And we did detect both mRNA and lipid sporadically, which suggests the fact that we are achieving mucus layer penetration with the therapeutic. And so if you advance to the next slide, Mara, these data, really put us in a position where, for one, we obviously want to make sure that we're completing our ongoing phase 1 and 2 trial, and really understanding everything we can learn from that, but also where we're really excited about being able, especially in the role that I've come in on as leading the pulmonary discovery effort, to be able to come in and really think about how we can even improve upon what we've developed so far. And so that really is what my team is focused on, is really thinking about what's our next generation platform that can really push us to an improved level of potency or an improved, administration profile. What can we do in order to really improve the probability that we're going to be able to develop a treatment that will have the type of efficacy that we're looking for. And what we've done, between Richard and myself, has really been to build out a pulmonary research capacity that actually includes all of these components, really making sure that we're understanding the biology, both from an in vitro, as well as an in vivo perspective, understanding, the cellular, but also the organismal. And recognizing that with this complex drug product, we also need to really become even better at understanding, things like aerosol science and being able to understand how we can optimize the delivery of this platform. So with that, Mara, we just have a couple of additional slides that are starting to provide a view of how we're thinking about what those next generation assets could look like. And when, Richard had, already commented nicely that these mRNA products really consist of two pieces. One is the mRNA, the other is the delivery vehicle. And we're looking at both of those and what we can do to really improve the potency for each one. And what's shown here on the right-hand side is a chloride current from an Using chamber assay that I'm sure many people here will be familiar with. And what's shown in this purplish blue line in this Using chamber current is really looking at our original codon optimized sequence and seeing what tricks can we pull in terms of coding the exact same amino acid sequence, but actually changing the underlying mRNA. Let's make wild type CFTR protein, but let's figure out if we can optimize the sequence that creates that. And in doing so, what we can see is a 1.5-fold increase from some early, assessments of some of these codon optimized variants. So now these are still encoding the exact same wild type amino acid sequence, but getting there via a different mRNA solution that will allow for increased amount of protein, increased amount of CFTR function for the amount of material that's delivered. And what we're looking at specifically within this plot are two different ways of stimulating CFTR that's shown in these first two arrows. And then subsequently in the last arrow, really confirming with utilization of a CFTR inhibitor that this change that we're getting is driven through CFTR function. So we'll continue to be moving these forward and assessing ways of improving the level of CFTR function that we can get by optimizing that payload component of our drug treatment. The other component that we've already spoken to, we'll cover here on the next slide, which is our last piece of data, is really thinking about doing everything we can in order to increase the level of, the potency of the lipids that we're utilizing when we're administering the RNA payload. And what's shown here is a method called immunofluorescence. What we're looking at is microscopy images from a mouse, where essentially we're looking at epithelial cells, and where this DAPI stain is staining for a nucleus. So every time you see one of these little blue dots, that reflects the nucleus of a cell. And what we're looking at in this orange is specifically the expression of a CFTR protein. And so what we're comparing in this slide between the top and bottom rows is either a saline control, where we're essentially just administering saline to animals, and where we can see our DAPI stain, everything looks fine, we can see the nuclei, but yet we don't see any CFTR expression. And when we deliver a lead lipid for NextGen encoding our CFTR mRNA, we can see not only with this middle panel that's detecting CFTR in the appropriate localization, that actually when we do that DAPI CFTR overlay, and particularly in the blow up on the right hand side, we can actually see that the CFTR is really localized in the apical epithelium, the top of these epithelial cells, exactly where we would expect CFTR to be expressing in its normal endogenous form, where it's able to be present on the top membrane, this apical membrane, and be able to carry out its intended function of transporting ions that will then in turn drive the appropriate hydration of the mucus and allow theoretically restoration of the cilia function, and in turn, essentially limit that pathophysiological cascade that results from CFTR dysfunction. So this is just a little flavor of the type of work that we're doing. It's a question not only of, continuing to advance this research so that we can better understand and better characterize additional delivery vehicles and additional cargoes, but also increasing our depth of knowledge by doing assays like this and some additional assays that really help us to understand, how to be administering these compounds, down the road as we advance them towards the clinic. So, it's really exciting for us. I hope that you can feel that, and I hope that you guys share some of that excitement. It certainly has been fun to be able to have an opportunity to speak to this group, and I'm sure I'm speaking on both Richard and my behalf when I say that.